Pradip already, Dr. Juthika introduced me. Thank you, Dr. Juthika, for such a nice introduction about me. Is the slides is visible to all?
Yes, sir.
Okay, and my voice is clear? Whether my voice is clear?
Clear, sir.
Yes, sir. Yes, sir.
Okay. Good morning. Good morning, all. How many participants are there?
Good morning, sir.
How many
Now,
participants are there?
63 has joined.
Oh, good number. Okay, good number. Okay.
Yes, sir.
So all are from faculties or some students are also here?
Sir, all are faculties.
Oh, it's okay. Okay. Thank you all. Thank you all. Okay. As Dr. Juthika giving me this opportunity and is in her institute. So this may be the second talk in similar type of series. Okay.
Yes, sir. Earlier was elementary and this one is advanced.
Okay.
Yes, sir.
So anyway, I do not know earlier those who have listened to my talk, okay, whether how many will be there, okay. So if some numbers are there to this talk, anyway, I will just, many materials will be common, but I will be discussing these things in different way, okay. So as decided, okay, in this busy schedule of the academic end of the sessions, okay, I just could manage to gather some materials from others and what about we are doing in our research group. Okay, so let's start today. Today's talk entitled is core cell polymer nanoparticles for the drug delivery applications. Okay, you see this is the campus, beautiful campus of IIT. Okay, let's say usually I show what we do. Already Dr. Juthika told these things, materials for healthcare and therapeutic applications. So we cover many materials. Okay, so from organic, inorganic, composite materials. Okay, and we use them. So research area we focus. This slide usually I show to know the audience what we are doing actually. Polymer based nanotechnology and biomaterials. Obviously we do work in the nanoscale, not in bulk. Okay, so and whatever I will telling also, this nanoscale delivery systems, we make delivery, we model them, we design them. So if you see the therapeutic aspects, so we have to take the medicines or some growth factors, insulin, anti-cancer drugs, immunomodulatory gas, gene therapy agents, vaccine. For the vaccine purpose also we have to take some immunity enhance the materials which will increase the immunity in this body. Let's say some MTB, little do all these things. So and we work recently. We are doing work on infectious disease. Okay, so what we do actually, we do synthesized. Okay, if it is polymer, biodegradable polymers and similar type, which is not infect the body. So ultimately be absorbed by the body or it will be go out from the body. So they have the various properties. Okay, biomaterials we can create in various functional way. Okay, they can actually recognize the drugs. Surface switching property can also be there. So biomaterials come carbon-based materials, biological applications you can use. We will show that this type of materials also can be used for the core cell purposes. Okay, so so conducting polymers, some sensing we made there. We will not show here conducting polymer core cell materials. Anyway, materials, polymer ceramics, metal composite, which will be mixed. Okay, so we functionalize them, porous, hollow structure, all these things will be there. So who work with us? So life science people, physical science, chemical and biochemical engineering, and let's say jointly we work. Okay. So since its slides is preventing from up, let's check now. Okay, how I'll remove this. Okay, so the concept of targets Okay, what have I have told that the target. Okay, let's say this. Let's say these things. There is a letters inside this letter. You will be putting some. Okay, there's an envelope will be there. You will put some letters. It will be closed, and there should be some carrier. So all together, let's say this is a system. Okay, this is a system. Letter is there inside the envelope, and there it will be a carrier like earlier ancient. Pigeon was the carrier. They took the letters and they gave this thing. So to the destination. So required person. Here, destination will be, let's say, for the infected diseases. Okay, so polymers or polymer-based materials will be there. One other concept of postal service for the targeted. Let's say you are sitting in Guwahati or somewhere else. Okay, there will be some postal box. You will have a letter. Okay, and you should write the addresses to whom will be the recipient. Okay, then you put, let's say, inside the letter box according to the pin number, destination. Okay, postman. Whenever it will be reached to the destination city, the postman sort out the things, letters, and they took to the destination or the address. So these are all the example of the target. So target letter. Let's say email service. Whenever you send us the email, okay, there is a defined address are there. Okay, you send, write, or you compose the email and then you send. So it will be, there should be recipient. Okay, so different type of targets example will be there. On others, okay, the mobile phone, mobile number, the number, this number addressed to a let's say allotted to a specified person. Okay, and you ring it, so it will be ringing a particular person's mobile. But with the mobile, okay, if you send email, most of the cases it will not, if you have written the address properly, this will not miss the target. Okay, it will reach to the recipient who has the password and this ID can open the email. So this is email is very much specific. But what is mobile happen? So mobile happen is the cross connection. Okay, so cross connection. So it is exactly similar to that medicine I will be telling. So if you have the cross connection, then what will happen? Okay, cross connection means you will find, let's say you wanted to call someone else. By cross connection, you will be, maybe from the opposite side, you will be getting some other voice. So this type of, so miss the target. Some cases miss the target due to the overlap and cross connection. So similar type of things occur whenever we use the materials, okay, or nanoparticles or drug delivery devices. So some cases, most of the cases, let's say they target to the specific person, but in the few cases they miss the target. So this phone number is a very ideal example that how the target we fix the target, but sometimes we miss the target due to the network problem or due to the environmental problem, all other things, weather problem. Okay, so now slowly I will be reaching, let's say, concept, how the concept has come for the nano. Nano is just very small, 10 to the power 9. The concept is
ನೀವು ಒಂದು ಕೆಲಸ ಮಾಡಿ, ಇದರಲ್ಲಿ ಹೆಸರು ಹೇಳಿ ನಂಗೆ.
some voice is coming from my side.
ಇದು ಅಟೆಂಡೆನ್ಸ್ ಬುಕ್ ತಗೊಂಡೆ, ಹೆಸರು ಹೇಳಿ, ನಾ ಮಾರ್ಕ್ ಹೇಳ್ತೇನೆ ಅದನ್ನ.
Okay, so what is that? The concept is very old. Okay, this is very smaller size, 10 to the power -9 in nanometer scales. Okay, so let's say working of lizard. If you see that they are thumb pads made up with polymer materials. Okay, it's a very ideal example. If you say that, if you see the feather of peacock, so they are made up also these polymeric materials, so biological polymers. Okay, they reflex and give different, okay, angle. Like a prism, they give the different color. So the way all others, let's say some inorganic concept. So nano, so usually whatever we will be discussing below 100 nanometers, this can be 1-dimensional, 2-dimensional, 3-dimensional. So if you accept these things as a polymer, so 1-dimensional polymer fiber, 2-dimensional, let's say flake will be there, let's say polymer film, all other things. 3-dimensional will be defined geometry. Okay. So let's say How small is nano? You can see this DNA, RNA, all other things. Okay. So nano, if you compare these things with the human hair is almost 10 to the power -5 or micron range. Okay, 3 to 4 micron. But okay, if you see the nano is 10 to the power -9. Okay, -9 in this range. Okay. Let's say if atom diameter is 3 angstrom, then let's say if you multiply by 10, so it is almost 10 times larger than the atom. But if you see a pin, head of a pin, so pin is 1 to 2 millimeter. So this is a bigger in size. But DNA, RNA, carbon nanotube, you see 2 nanometer. Okay. So these are the small, so small. So constituent of, let's say, will be coming, these materials will have the uses for making the core cell materials. Okay. So metal, whenever you will be the constituent for the nanocapsules, okay, if you use the inorganic materials, you have to choose also biocompatible materials. I will be telling, let's say, inorganic metal, ceramic, oxide, polymer, different type, copolymer, block copolymer, composite, metal polymer, you can make it, polymer, all little example I will be giving. Non-metal, semiconductor, fullerene and other composites can be there. Another type of is that you can conjugate the virus with the polymers, and that cases also you can make the virus polymer core cell materials. So I will be telling all the definition. And carbon materials also, graphene oxide. So this can be there. Okay, now example, let's say, how is these things? Let's say if you take these inorganic materials, okay, there will be, let's say, zinc oxide, copper oxide, titanium oxide. There will be various sizes. Okay. So whenever all together, they will be there inside this polymer core cell materials. Their property also tune the overall. Okay. And if you consider the organic, let's say mycelis, liposome, hybrid, dendrimer, all others. Let's say myceli, liposome, hybrid materials, they are made up of lipid or protein type. So what is property of them? They are the self-assemble the mycelis. Okay. The long chain fatty acid, okay, they will be having the hydrophobic chain and hydrophilic chains. Okay, according to this their property, they will assemble and critical mycelial condensation concentration. Lipogen also similar way. So what you see that the head they are making the outer wall, and inside this chain will be there, tail will be there. So I will be telling how it will be making is a core cell. Okay, lipogen similar way. There will be a wall and there will be hollow space. Okay, hybrid materials also. Dendrimer also a porous material, so made of polymer. So what is the difference? You say nanosphere usually solid, but nanocapsules having the hollow core will be there. So fullerene is hollow, defined structure will be there. Let's say. So now let's say we'll be functionalizing. Basic just parameters I am telling. If you would like to functionalize these things, various type of pores, you can, different sizes of pores. Okay, they have some nomenclature. Mesopores 2 to 50 nanometers, micropores 2 nanometer. Okay, macropores above 50 nanometers. Okay, so how you can make the porous structure? So there will be the way different how it look like actually. These all are almost, other than the gray picture, all are the FM picture. So you can functionalize in various way and various dimension. Okay. So what is polymeric biomaterials? Biomaterial science is the physical and biological study of materials and their interaction with the biological environment, and they will not create any allergic manner. Okay. So they will not react, or if react, whatever the products will be there, they will not harmful for them inside this body. Okay. Polymeric biomaterial, what are polymeric biomaterials? Okay, let's say biomaterials is the non-viral materials used in medical devices intended to interact. Okay, this will be there. So properties, what will be the properties? Properties of nanobiomaterials, let's say polymer cases, they will have some physical property, they will have some chemical properties, they will have some biological properties. So if it is polymer, their strength, tensile compressive strength important, elasticity important, architecture also important, and pore I have discussed. So chemical property, biodegradability, water content, toxicity, everything will be many important. And biological property will be depend on what moieties are there, how the functionality is there. Okay, what polymers you have taken, what is the MAR you have taken. Okay. Release of biological component will be depending on the moieties. So what moiety? And lifetime is Very important means how much time they will be withstand inside this body. So all these things, whenever we will be designing these core shell materials or any type of capsule, that is important. Okay. So let's say target, what we mentioned the target. Let's say you will have, okay, you have germs capsules, and this is the target, let's say infected cells. Okay, infected cells. Then what will happen? So that will be go to the infected cells. Okay, that is the target. So target is this, and let's say this will be this. So these are the your core shell polymers, let's say. Okay, now you see chemotherapy cases also it's happened. You have a capsules. Capsule have lot of medicines inside this capsule in the hollow space, and that will be killing the cancer cells. Okay, let's say this is the cancer cell. But whatever the I have given the cross connection of mobile number, if they miss the target, then they will kill the normal cells also, which we usually don't desire. Okay. So what other advantage is that why we will be using this thing, this type of core cell materials or other things. So inside the core cell, okay, we can take lot of medicine, which actually will circulate inside this body. Okay. Inside the body means so many times. Yeah, okay, inside this body it will circulate. Otherwise, what will happen if you take the free drugs? Okay, it will be direct contact on the skin, let's say, okay, and sunlight. Okay, no indirect contact, but sunlight will be passing. Penetration depth will be there. So effect of light will be there. Not safe in biological fluid because most of the cases in the sunlight they react, and if you take free, that by through the urinary tract they can go out easily. Now see. So whenever we are taking this type of materials, they translocate. Okay, if you inhale or if you take this capsule type of thing, so it will go to stomach, it will pass. Okay, or if you take the injection, it will go each and everywhere. Everywhere. Let's say kidney, urinary tract, fetus, heart. Okay, brain, liver. So everywhere, even cells inside the cell membrane. So that will be there. So they will have the smaller size of property, which actually help them to reach to the target. Okay, so how the size is required? If a tumor is there, enhance uptake for particles with size between 30 to 200 nanometers. Okay, let's say heart. Okay, 30 to 200 nanometers. Normal blood stream, you can take any size. Okay, in nano. Liver usually 20 to 150, or hepatic filtration for the particles 10 to 20 will be there. Okay, 10 to 20 size will be filtrated. So high you have to take 20 to 50, above 20 nanometer. So how we will be deciding which? If you target in the spleen, let's say spleen, okay, rapid clearance of particles with size larger than 200 nanometers will be there. If it is kidney, then clearance through the excretion for the particles of size 8 nanometers. So you have to take in, okay, more bigger than. So how you see how the nephron and other things will be there, how the nanoparticles will be translocated through the kidney, because every time, let's say, it will pass to the kidney. Okay, filtration system will be there. So you have to decide these particles. No. Now why nano is important for biomedical application? Let's say if bigger sizes of particles, they will circulate through the bloodstream. Okay, they will not pass to the cell membrane. Okay, they will circulate. But if there is infected cells, okay, then what happen actually? Usually the gap create in between the infected cells, or body is infected due to the parameters, change in the parameters inside the body. So there will be endothelial cells where the gap will be created, and according to your size, nanocapsule, core cell, whatever will be there, they will pass through this and they will be reaching inside this, inside the infected region. Let's say here I have shown the tumor tissue. So 100 to 800 nanometers gap usually create when the cancer created. So below that particles, if you take, it will not go through this channel. But if you take larger size, it will be always, it will circulate through this thing. Okay. How nanomaterials reach to the target? There are many mechanisms are there. Let's say SR mediated. Okay. Mediated phagocytosis, pinocytosis. There are a lot of biological mechanisms are there. Caveolin mediated endocytosis, okay, Catherine mediated endocytosis. So depending on the size, shape, charges, okay. So scavenger receptors means you have to have some receptor which your polymers or whatever will be there, they will receive by this. So mechanism will be there. So different barrier, whenever it will pass to the barrier, let's say is reached to the cell membrane. Okay, so they will be inter. What about this? I have told here the mechanism. Then what is inside the cell membrane in the cytoplasm? You see, lot of pH changes will be there. So polymer actually face lot of problem because it will have some functional property and in the organic material, the Golgi body, mitochondria, okay, nucleus. You see that various places and the various region in contact region. Okay, there will be pH will be different. So that very important. What others is that, let's say you will be using this core cell materials, okay, polymer material, surface cells, okay, of the cells, okay. The cell surface usually made up with organic components, OH, protein, peptide, lipid, okay, any others, so many things. So whenever these materials, polymer materials will be there, we have to design in proper way that they can cross this barrier easily, or there should not be so much barrier to pass these things, okay. But most of the cases it become very difficult due to the charges. Okay, now how can we proceed? So we make nanosize, porous, hallomesoporous, functional property, need to functionalize, drug, memory, need to function in our capsule for drug and gene delivery. All these things will be there. Why we need actually? We need to load huge amount of drugs, okay, and there should not any uptake problem or loading problem or drug or genes. Okay, whenever loading problem will be that transfer also will be there inside this body or infected cells. And you have to decide the uniform sizes, have to make where you can make the doses properly, okay, and selectivity problem should not be lost. Okay, just a minute. So selectivity problem will not be there now. So how you can make this polymer nanomaterial? So if I believe that many of you know, you can make precipitation, suspension, dispersion, chemical routes, emulsion, mini emulsion, or microemulsion. So emulsion method are very much useful to make these polymer capsules. Okay, now I will be coming. So what parameters you can control? Monomer size, stirring speed, monomer droplet size, stabilization, solution stabilization, solvency continuous phase, or whatever these things, multiple surfactant you can also be used. Now see, what is, I have told so many things, but what is core cell? So core cell, let's say this is a spherical thing. There is a core, which is really the central part, okay, and there is a cell. It's like a egg cell, okay. So you see, cell most of the cases the solid, okay, cells are solid, but core what can be there? Core can be solid, core can be liquid, core can be gas. So when you can put the gas here, if it is empty, okay, on a hollow core. So core can be hollow or solid, okay. You can put liquids here, or it can be there liquid, okay. And cell, whatever this core is there, cell will have little bit solidic property, okay. So cell always should be solid, but core can be solid, liquid, gas, empty, okay. So this is actually core cell. So there will be a core and there will be a cell. So whenever you will be synthesizing, then you can vary the core size and cell size also. That is in our hand actually. That is in our hand. We can make by changing the process parameter. Now, so this is the core cell. There is a core and cell. Now, variation of core cell structure. Now see, okay, synergetic catalysis use will be there. Let's say this is the core cell. Template free approach will be there. We can use template free approach. Ultrasonic spray pyrolysis. These are the method how we can synthesize. Microwave polyol methods are the sieve in the bottle method. So there will be a lot of methods since the time is not there, but for the information, this method you can use. Okay. So synthesis type. So what can be the type? You can synthesize, let's say polymer core cell nanostructure, silica core cell you can make, polymer core cell, polymer core cell, magnetic core cell means magnetic materials will be there, the core, and cell can be any type of material. Monodisperse cores, so uniform size of core can be there, okay, cell in nanostructure. And bimetallic core cell, if you taking the metallic, so outer surface can take metal and inner also core can also be taking the core and cell both are will be the metal. So you have in your hand, okay, you have in your hand. Now see core cell nanostructure, how can single core cell nanostructure, let's say one core, one cell. multiple core, single cell. So there is a cell and there can be multiple core can be there, bigger size. Okay, you see multiple core, single core and multiple cell. So there will be a single cell. Okay, single core, but layer by layer deposition of the cell can be there. So this is useful when you wanted to make the functionality of the cells. Okay, how you wanted to use. Now you see multiple core and multiple cells. So core different and cell also different. So core materials can be different, cell material can also be different depending on your choice in which purpose you wanted to use. So in those two cases, very important are there, or in these three cases, let's say multiple core in single cell, you can use multiple types. Multiples mean multiple types, not multiple types and multiple size. And these cases also, whenever cell layer are there, let's say different polymer. First, you can take a layer of one polymer. Second, you can take a layer of polymers. Second and third type of also you can take. And let's say in these cases, fourth cases, so core can also be with the different materials and cell also can be different materials. So you have in hand. Okay. So lot of things you can play. So here we will be discussing application, drug delivery, other than the drug delivery, bioimaging, sensor, environmental, photovoltaic, solar cell application, lot of. So we will be defining, let's say, a drug delivery, bioimaging. This purpose we will be defining. Now say core cell preparation of polymers, how you can. So let's say you have a monomer 1, you have a core, okay, and monomer 2, okay. Let's say monomer 1, you make a core, okay. Okay, you take second type, monomer 2 means you have to polymerize. So polymer will be there inside the core and polymer will be there inside this cell. But you have to choose that in such a way that there will be an affinity or electrostatic interaction, okay, will work between the monomer 1 and monomer 2. Even the monomer 1 produce these particles, but there will be interaction force at least, or affinity should be there with the monomer, so that monomer can deposit on the surface and then it can create the cell. Otherwise, what will happen if it's monomer to create the repulsive force on the surface of the cells, then a surface of the core, then what will happen? This monomer 2 will not be deposited. So this is always true for other cases also. If you take metals, different composite materials also core cell, so there will be gap will be there or repulsion will be there. So if there is no affinity, then monomer 2 will not deposited after formation of the polymer. So this is the basic principle you have to keep in mind. So whenever you will be, let's say, preparing the core cell, okay, and let's say your aim was there preparing the core cell, but you found that the monomer 1 produced polymer 2 and monomer 1 produced the polymer 1. So it's not making the core cell. It is means that they are not compatible. Either one is hydrophobic, one is hydrophilic, or both are in opposite property. Ionic charge, if opposite, then they will be deposited. So ionic charge also can interact. So if one is cationic, another is cationic also, that cases also will not, deposit will not be there. So monomer reactive surfactants, let's say one surfactant will be there. Emulsion method actually this happen. The surfactant molecules usually produce the droplets, okay, center part. And on the droplets, if droplets are made up of, let's say, high molecular weight, long chain fatty acid, okay, so they make the droplets, and on the droplets, okay, on the droplets surface there will be opposite charge which actually attract the polymer chains and allow the deposition layer by layer on the droplet surface, okay. So then these cases, so it will be liquid. So monomer 1 and monomer 2, you take monomer 1 producing, let's say, in the opposite, you see that how it is happening. In these cases, monomer was the central part, and these cases will be monomer 2 will be the second part. But you see, monomer 2 producing core, then monomer 1 making the, let's say, small particles. Okay, that particle will be deposited here, and further polymerization, layer by layer deposition also will be there, and it will be a smooth layer will be forming. How I will be showing example this thing. So amphiphilic block copolymers, what is amphiphilic block copolymers? Which have positive negative charge. So one have one size, okay, it's called also deuteronic. So one side of the chain will be positive negative, another side will be negative positive. So both will be dominating. So block C, let's say micelle will be there, that amphiphilic polymers will be attached on a block micelles. Okay, then again the polymerization will be occurred by the cross-linking agent. Okay, so they make a cross-linker. I will be showing, I'll see actually example is there. Okay. Okay, so on the block C micelles, the cross-link polymer will be forming. Okay, so cross-linking will be forming, and core shell cross-linking will be there. The core will be there, and core shell similar type of the third type. The cell will be generated, and layer by layer deposition of the amphiphilic block of polymers will be there. So these are the actually how to make. So core cell preparation, inorganic polymers, let's say example, okay, polymer cells and silica, you take core. What is the size you will be taking? 300 to 1000, let's say you are taking, and you will having the polymerization method, okay, and polymerization will be there. So the silica will have negatively charged OH, okay, that negatively charged actually attract, okay, the polymer materials, okay, so attracts and allow the deposition. Now you see that. So whenever you will be changing the core diameters, it is means that core diameter, if you would like to make different, then you have to take the different in core diameter. Okay, you can vary. So silica is a material which is very easy to make in different sizes. Okay, let's say from 10 nanometers to 100,000 nanometer you can make, and according to your use you can put during the reaction. Okay, so core and allowing this reaction core shell. Now you take out the silica, then will be hollow materials will be there. So that cases hollow will be generated. Let's say core shell polymers with functionalization. Let's say silica coupling agent, you have to use that. It will be coupled on the surface of the silica. Silica will have the opposite negative charge. So if you take the organic moieties, okay, then what will happen? Easily it will couple, okay, with the negatively charged OH group of silica. Okay, then you allow the surfactant or polymers. Okay, so if you take the surfactant will be attached on the surface, and then you take monomer. So monomer will be polymerized by free radical method. Let's say you know the free radical method, I will believe. Okay, by free radical polymerization method, let's say addition polymerization, it will be polymerization will be formed, and due to this, okay, coupling agent, they will be allowing the deposition of this. Okay, so you see that there is a core, there is a coupling agent also will be there, and surfactant or polymerization. So it can be there. Though above the surfactant they can make the layer, or they can cross the surfactant, and on the surface also they can make the layer. So usually surfactants are used to control the sizes of the particles. Okay, you have to keep in mind. Now, inorganic and polymers. Okay, let's say silica is there. Amine functionalized tetraethyl orthosilicate will be there, which usually you can functionalize these things. So this, why I am taking the example of silica is this. Silica is very easy, commonly used. You will get maximum number of papers where the core cell materials have been prepared with the silica. Okay, amine functionalized. So aminopropyl tetraethyl orthosilicate. This is the name of this APTS. So amine functional group is there. Then helical polymers, you take that. Okay, what about if these things, that there will be a functionalization NH group? Let's say if COOH, NH are there, they will easily couple. Okay, so there is a polymer layer easily will be there. So you have to choose is that what you want. Now see, now real picture you see, sensitive parameter for the size. So shape size depend on the molecular weight of the polymers. Okay, how you are preparing. Temperature also will be controlling. You see that with temperature size can be there. Okay, and during the preparation also, if you take the solvent, different solvent, different sizes also will be forming. Okay, surface can also be tuned. Okay, with surface have very important property actually for biological purposes depending on the size. Let's say smaller size, this is 40 nanometer inside, so it's a smooth surface. What about the roughness is there? That is only due to the folding of the chains. Okay, that is only the folding of the chains or the atomic height. Okay, atomic height. But second case is larger particle, polymer chains coiled in bigger manner. Okay, so roughness will be very much different. And the bigger cases, whatever I have shown, the polymer particles deposit on the surface, and that cases roughness will be there. So whenever you will be loading these drugs, so interaction of the drugs and this outer surface is very important, and interaction of this outer surface and the cell, whenever they will be going inside the cell or interact with the cell membrane, that is very important. So these parameters all. Now, for example, you see the smaller particles, they come closer to each other. Okay, they will assemble, and after the assembling, in the second stage, they will be forming layer by layer deposition. Okay, this is our work actually. So this we have shown how it can make that. So whatever example I have given, that was the other work, but this is other work, the resultant. Okay, okay, and you see that whenever the larger sizes of particles is coming, the roughness actually again is increasing. First is it increase, okay, then increase because of the layer by layer deposition. Then what happened actually? What happened actually? Whenever you take the polymer particles or polymer capsules, you can load the drugs molecules. Here it is shown that fluorescent drug molecules, okay, attached. Now you see that. So from the surface, it is coming very deep, and intense green fluorescent. But from the center, it is not coming. It is black. It is means that the drug molecules are attached on the surface. Okay, and core may be solid. Okay, its drug cannot be penetrated. Okay, so the what result is coming, conclusion can be coming. If your core is solid, okay, then drug molecules will be can be loaded on the surface, or it can have little penetration according to the thickness of the polymer cells. Okay, so you have to keep in mind that thing. This slide maybe I have shown earlier, but I have explained differently since we are discussing the core. Okay, so core solid means your drug molecules will not go penetrate up to the core. It will be there on the cell only. So according to the requirement, let's say you don't want your drug molecules will be there at core, because if it is there inside the core, it will take time to release. Okay, so I will be telling there is a term, it's called burst release, which is immediately release is required. So these things. Now again, you see in the better way. So this is the hollow core actually. This is a hollow polymer particles due to the hydrophobic and hydrophilic interaction. Okay, only on the surface drug molecules are attached. Let's say fluorescent molecule. So you are actually, advantage also is there, disadvantage is there. Disadvantage, what is that? Let's say there are a lot of space, but you are unable to load the drug molecule. This may be solid, or there will be some electrostatic interaction which is not helping the drug molecules to go inside it. Okay, so according to the requirement you can make, but you have to keep in mind when hydrophobic surfaces are there, you can load the hydrophobic drug molecules. Now you see this real example, which there is a polymethyl acrylate capsules. Okay, a coat has been taken by the oil. Okay, oil droplets, and on the oil droplets, layer by layer deposition of polymers. Okay, so hollow cores, but not really hollow, the liquid are there. Okay, PMMA capsules. So oil plus drug loading. Then you have to load in some liquid way. Solid drugs cannot load. So you can take the oil or something liquid. So which will actually take by the diffusion method or some electrostatic interaction method. So inside this core, finally, drug particles will be there in the liquid. Okay, then you can solidify. Okay, if liquid is there, then they can diffuse from liquid to liquid. But if again it's become solidified, then that cases what will happen, release will be very less. Okay, release will be very less. Why? So you have to, liquid will go inside, then it will dissolve, then it can. So solid particles cannot come out. So it will be in the solution mode. So this, how you will come to know whether how is this? You see that drug molecules are functionalized with rhodamine actually. This is rhodamine is a fluorescence molecule. It's give the red fluorescence. So it's giving intense fluorescent red throughout the capsule. It means that not like earlier. So it's penetrate, drug penetrates even in the core and the cells. Okay. So this you can challenge again the infected cell, and whether whatever I have told, this has gone to the cells or not. Okay. So this will go inside the cells. If so, then you will see the red spots. So same you have to do the fluorescence imaging. Okay, by confocal microscope or fluorescent microscope. Okay. So these red are coming this. Okay. So these are loaded. Fine. So why porous structure is needed? Okay, why porous structure is needed in core cell materials is that, let's say you wanted to ibuprofen or gentamicin, this alendronate, okay, erythromycin, these are the drug molecules. So if you deliver these drug molecules, whatever example is given, actually these all are the almost all are the painkillers, and whatever the antibiotics are there, they have the side effect. Okay, so if you take these drug molecules inside these pores, then what will happen? They easily cannot go out. Okay, they have to require some time to go out, or other cases the pores will be some wall which is made up of solid or polymers, whatever is there. Okay, so they will be actually preserved inside these pores. Okay, so easily they will not react. Bovine albumin serum also, it's similar way. Okay, and other cases, if you take cage as a cage pore, so inside this case the drug molecule will be safe. Second cases. So why need that? Why need this pore of the materials? Because let's say pore is one type of pore which is normalized material, very slow drug release with oral zero kinetics, closely related to non-functionalized. These cases. Second cases, very common non-functionalized pores. Second cases, if pores are non-functionalized, let's say zero hour kinetics, and it will follow the exponential. Okay, so high surface area, small pores. Third cases, okay, C correspond to a zero order kinetics to those release process which are only time dependent. So this is very much acceptable. Okay, zero order, but with time continuous release will be there. It is not like the slope will be changing. So continuous slope. It is means that if you take the drug molecules inside this core or whatever inside these pores, the continuous dose will be released. Okay, other cases will be there. These cases is there, the functionalized nanoparticles, drug molecules will be attached, but until unless you giving the thrust, let's say magnetic field, temperature, changing pH or electric field, until unless this type of threat is giving, the drug will not be released. So once threat will be giving, drug will be released, and let's say you are taking this stimulation, threat or stimulation, then it will taking out, then it will stop. So in every thrust, okay, drug will be released like this. Okay, in every thrust, drug will be released like this. Okay, so every type of functional properties, core cell materials will have the different property. Now diffusion and degradation will be there. Let's say this is the core cell materials which are hollow, and whatever I have told that what is the advantage of the core cell materials, you see this. So inside this core, hollow space is there where drug molecules is there inside the liquids. Okay, so the outer surface is the polymer. Okay, this polymer should not soluble. If it is soluble, what will happen? Structure will be destroyed. So insoluble polymer membrane will be there, which can be the porous or like porous. Okay, so then diffusion will be easy. If it is not porous, only the polymer networks are there, what will happen? Diffusion control will be there, but until unless, okay, so until unless the something thrust is giving, okay, they will be encapped. Okay, because if both the cases liquid are there, then diffusion is easy. Otherwise, inside the cross-linking cases, they will be entrapped. Okay, they will be entrapped. And with respect to the second cases, here less amount of drugs can be loaded. Okay, so if you are loading, let's say one capsule, if it is has huge number of drug molecules, okay, or medicine, then what will happen? After the delivery, it will give sustained release for longer period of time. Okay, and if it is solid, then what will happen? Small amount will be there and less number of drug molecules will be there. So drug delivery period will be very less. Second cases is there, it's a completely solid. Okay, if you can compare, completely solid, drug molecules are entrapped properly. Okay, then release in these cases will not be so easy until unless there will be a degradation. So until unless there will be a degradation of polymer chain, okay, until unless degradation is there, no drug molecules will be released. Okay, so now I think it is very clear that when you taking the core shell hollow type of these capsules, what is the advantage is there with respect to the solid or networks type of these there. So this is the advantage. Now other cases is that there is the core shell prepare here, let's say from micelles. If you take with the pH sensitivity, with the in terms of the pH sensitivity, so whenever pH either increase or decrease, so there will be some effect, there will be some agitation inside this cell assembly structure. Okay, then the network will be break and the drug molecules will be released. Same cases, depending on the structure also, if you increase or decrease, similar type of phenomena will be observed. For the liposome cases, so this actually happen in the mycelium. So if it is there in liposome, liposomes actually it is like membrane of the lipid or peptides, okay, where lot of channels are there through which the drug molecules can enter. Okay. So if pH or increase or decrease depending on the structure, then they will expand. Okay, channel will be increased and they will be allowing to go out the drug molecule. So these are these. So hydrogel cases, this is solid cases I have shown. Okay, this is also one type. Since it is not related, this is not hydrogel, is not a core shell, it's a porous structure, let's say, but it will helpful for this. Now, let's say sensitivity is temperature. There will be lower critical solution temperature. At that cases, you can easily load, and when you will be increasing the temperature, then again the structure will be expand, pore will be increased, and from the hollow core, okay, with increasing the size of the pores, okay, the drug molecules will be going out. So lower critical solution temperature and lower critical solution above the lower critical solution temperature. So what is lower critical? Where the secondary phase at the temperature, at the lowest temperature, where the secondary phase generate. Okay, so that is the thing. So in these cases, there is a core, the polymers, and then let's say brass type network is there on the surface. Inside the brass or polymer chain network structure, you can entrap the drug molecules. Okay, and if you here the sensation is the temperature. Whenever temperature sensation will be there, these brasses, polymer brass on the surface, okay, they will be change the network structure. They will vibrate. Okay, and let's say there will be secondary emission of the light are there, or secondary agitation will be created, but vibration will be created, which will help to go out the drug molecule. So these are the advantages of the core molecule. Photosensitization case also. So if you have these things, core cell materials here, mycelis or whatever you are taking, if it is sensitive to the light, okay, then what will happen? It will allow to release. Okay. Other cases, it will be there inside. So, or it can release only, it can make the cleavage, or it can destroy. Okay. So these are this use of hydrogel. So I was telling that. So why you will be use it for making the core cell materials? What is the role of hydrogel? Okay. So let's see what is hydrogel. It's you can have a linear polymers. Okay, like this, which are functional group. Let's say acrylate you have taken. You can radicals will be forming. They will be attaching. Okay, together, and new marks will be created. That marks actually attach on the surface of the network. Okay, then cross-linking polymer chain will be there. So if you have a core, then on the surface of the core there will be hydrogel. So what is the hydrogel property is that whenever you will be taking inside the water, it will expand. Almost 90% volume will be increased, and that volume actually increasing volume will help to release. Okay, if it is not released, if it is there inside this core, then it will, you have to give little bit sensation or thrust. Okay, so network formation, network structure advantage you can make above the surface of the core, let's say solid core. So this is secondary advantage will be there. Now there is different method. These nanoparticles you see, nanoparticles will be there inside this core. Okay, core I have shown multifunctional core will be there. And inside the network, this core will be there, which actually hydrogel network form. So, okay, so inside the core, you can load the medicine, and inside the hydrogel also. So if drug molecules is there hydrogel, there will be different release. And if drug molecules, let's say second drug molecules is there inside this core, they also will be released, and you can stimulate them. So how you can stimulate? I have already told: light, temperature, electric field, magnetic field, precursor, ionic strength. Okay. Everything can be there. Okay, so they will be released. This usually used for the wound healing. Okay, where let's say implantable materials, nanoparticles, implantable materials form. Let's say this is a metal where this is the muscles where wound created. You take the hydrogel based core cell nanoparticles, you fill it. Okay, with the medicine. So after filling, there will be lot of sodium ions, ionic. Okay, and they will help to addition of this tissue here and allow to grow this tissue. Okay, and they will fill the gap. Okay, so after some time what happened, since it is a hydrogel, it will allow allow the transfer, mass transfer not only the liquid, mass transfer of the blood flow by blood flow channel. Let's say vein will be created here, and they will allow to make the passages, okay, fluid flow, liquid flow, or blood flow passages. Okay, so slowly it will heal the wound. Okay, so the calcium ion will be there. If bone damage, then they will allow, this hydrogel will allow to diffuse the calcium ion, and they will grow the bones. So this all are advantage. Another thing is that hydrogel, let's say one example I have given, made up with polyethylene glycol, polylactic acid, polyacrylate. This type of hydrogel network will be there, and they can easily degrade in water. So if they degrade, they again will be forming polyethylene glycol, lactic acid, polyacrylic acid. If you see that polyethylene glycol is very much biocompatible, and lactic acid, this lactic acid, when generate, if it is gathered inside this material, lot of muscle pain are there. Okay, but what happened? Lactic acid, whatever they produce, they take part in the carboxylic acid cycle inside the biological system, and it is useful. Okay, so this is the use, and they will have different time, okay, required for the equilibrium, mass swelling, degradation. Okay, that's also advantages. Okay, let's go. So now see core cell polymers, whatever we have made. Okay, you will see this core cell materials. I will see. We cannot see. We do not know. This is the ACM images. You can see that nano sizes of particles almost 100 to 200 nanometers. Okay, almost monodispersed, but we do not know what is in this inside this core. Now we see. So size distribution, you see that the core are there. Okay, first we made, this is actually solid cores. Okay, then you see here the blacks. Okay, this is the high resolution TM image, 100 to 110 nanometers, 150 nanometers. And if you see that the thickness of the cell is 19 nanometers, we could achieve. Okay, and cell are there. Okay, so you can make different sizes of particles. If you see here, what you will find, there is light gray color is the cell, then solid, deep black is the silica particles, and then the liquid layer also is there. Okay, so liquid layer means this is the gap. Okay, nanoparticles of silica 100 to 110 nanometers, and there is a liquid gap. So if you can take out these things, I will also show it. Okay, so if you take the different ratio of the whatever I told, different ratio, you can take the polymers, inorganic, okay, nanoparticle. Instead of silica, you can take everything: iron, gold, silver, okay, any type of inorganic. So actually solid core always helps better to make the concrete structure. Okay, and it is sometimes easy, easy to remove. So by etching by sodium hydroxide or the HF, okay, hydrofluoric acid, you can remove these things. Now you see how it's looking like in the high resolution TM. Okay, this is the capsules. Thickness is 20 nanometers. What is thickness? Cell thickness is 20 nanometers, and the hollow cores. Okay, fill up with liquid. Now you see this core cell polymer nanoparticles, where this is the core. Okay, 5 nanometers, almost 5 to 10 nanometers. So you can vary, and it is full with liquid or let's say empty. And these cases, you see, this is the particles of let's say 100 to 150 nanometer. There's channel, number of channels. These are the channels. Actually, 1 2 channels means this is the passage. Okay, through which it can drug molecules can enter, or it can go out like a door. You can enter through a door, or if window are there, air can come out, come in and come out. Okay, so there will be a mass transfer channel. These cases I will be showing in high resolution. Again, further high resolution, the white spots are there. There is a pore, but there is a channel. Okay, pores are also is there, channel also. So there are two different type of advantage will be there. So making the pores and making the channel. Okay, so you see. So pH sensitivity. Let's say this hollow core, hollow here it has been showing like a key. Okay, when pH is there, low pH is there, they close this pore. Okay, close this pore, and above this pH, they open the door of this channel. Okay, then what's happened? So in this below pH, then you can load easily. Okay, or let's say it is opening or something happening. So drug molecules will be going, and it will entrap inside this core. Increase the pH, so drug molecule, then what will happen? Size will be expand. Okay, then drug molecules will coming out. So in these cases, it is entering, and these cases is coming out. So what is showing the clock? So pH up to 14. Okay, this is a pH clock. Okay, pH sensitive material, pH clock operating mechanized nanopolymers. This is this. So pH will be mechanized this type of core cell materials to loading and releasing. So first we have to design loading and releasing mechanism, then delivery things will be coming later at the target space. Now, so use other things, glucose sensitive. Let's say this is made of, I am not going to details. This is made of the hydrogel. Okay, this is made up of hydrogel, where the core are there lot of silver nanoparticles inside this core. Okay, and drug molecules are there inside this. So that's a different property. So what is this? These drug molecules, whenever I am taking, let's say insulin. Okay, glucose responsive is okay, and they have taken the photonic crystals. This has been done by some others. Photonic crystal zinc. So they actually give the light sensitivity. Okay, they radiate. Zinc is fluorescence material, so radiate the light. Whenever light will be radiate, they will excite the silver and drug molecules, and the glucose molecules will be coming out. Let's say somebody has high glucose content inside this body, and you inject the insulin loaded these capsules, and you give the fluorescence intensity. Okay, somewhere. So this will be region. So input glucose level, then response swelling, then output or optical signaling, insulin release will be there, and feedback will be there. So again, you take out the fluorescent light, then it will be quenching will be there, and it will prevent the release. So these are these. So specific glucose to, okay, SPR signal. Okay, surface plasmon resonance signal, transduction at physiological pH by molecularly imprinted responsive hybrid materials. So molecularly imprint means these are the actually chiral molecules. Molecules are attached with this structure polymer capsules. Okay, where the we have taken some others were taking the glucose molecules, insulin molecules, and silver, which actually give the light sensitivity and use for the glucose sensitivity. Now, other example, temperature cases, responsive cases. Actually, why it is important? Our body, let's say, have some fever. Okay, so fever, whenever increase the fever, that case is what will happen, or decrease the fever, what will happen? Let's say there is a core. Okay, where on the surface of the core some polymers are functionalized like this. Okay, functionalized polymer. Then core and cell will be actually network structure like this, made up with pH and NIPOM. Okay, NIPOM is a temperature responsive polymer which is attached together. Do you see? First case is core, then case is NIPOM, and then PEG. So what happens whenever body temperature will be increased, they will expand like this. Okay, and whenever they will decrease, they will contract. Okay, now see, at lower temperature, let's a little bit increase the temperature, and through the passage the drug molecules or whatever this, the medicine will be there. And let's say increase, let's say in the fever cases, when the body temperature, oh, I forgot to show something. Okay, then what will happen actually? Okay, these cases, drug molecule will be coming out from the core. Okay, drug molecule will be coming out. So here shown the doxorubicin, which is actually chemotherapy drug used for the anti-cancer therapy. So a lot of polymers you can functionalize on the surface of this. Okay, with respect, if we tell about the core cell structure or core cell polymer material. Okay, so core can be designed, cell can be designed, and you can see. So what further is that? Let's say there is a core, you functionalize with some other things, and you functionalize with DNA, RNA. Okay, ultimately you will have a functionalized polymer network chain where the DNA, RNA molecules will be there. So that you can actually deliver, okay, to the cells or the infected tumor sites, where actually it will help to kill the tumor. Now, let's say one polymers is there, polycaprolactone. It is used for the tissue engineering, but what we have done, we have made this type of capsules where hollow core, porous structure. You see, porous structure is there, hollow core, pore. So cell is porous, okay, and thickness is almost, let's say, 20 to 30 nanometer. Now, how is that? How you can make? You take the silica, okay, in the matrix, in the solvent form. Take the polymers, okay, take some surfactant. Then what will happen? So surfactant will be attached with the polymers, and along with the surfactant micelles, they will be deposited on the core like this. So red are surfactant, polymer network structure like this, okay, and silica is like this. Okay, now second cases, first you remove the micelles, then what will happen? Core will be generated, and third cases you remove the core. So you remove this core, then hollow will be there. So soft template is the micelles here, hard template is the silica. So whenever you will be taking the hard templates, okay, hollow core will be there. When we will be taking out the soft template, then mesopores will be there. It is like that, layer by layer deposition, like layer by layer deposition will be there. Okay. So if you take in the solvent some drug molecules, you can take. So hollow capsules will be generated. So we have published in somewhere else. So what in purpose we can use is that, okay, cell, we found that cell viable. We take the cells, normal cell, let's say here we have shown the skin cancer cell, they are very much viable. Okay, cell in the normal microscope you cannot see the particles. Only fluoro nanoparticles will be showing in the high resolution microscope. Okay, okay. So it's loaded with rhodamine, checked cell viability, it is very much viable. Here you can see a little bit, okay, white are cells and nanoparticles are actually coagulated, no problem. Okay, and then after loading the siRNA, it's challenged. Now see this cell, how the morphology has been changed. It is means that the siRNA loaded nanoparticles interact with the cytoplasm, DNA, okay, and then destroy the cell wall, okay, nucleus. They kill this. The killing efficiency with time, okay, will be increased. It is showing the green fluorescent intensity is decreasing, means cell is killing. Okay, now you see that. So PCR loaded with this, how the cell viability actually decrease. Okay, viability decrease means increase the cell death. Okay, if you take free siRNA, what will happen? It will attach on the surface of the cell only. If you take with the nanoparticles, then they will be going even in nucleus, in the cytoplasm, everywhere they will be going. Okay, you see, actually here siRNA are tagged with EGTF. Okay, extra green fluorescence protein. Okay, so why is green fluorescence protein not showing these things? So it has gone. So what actually happened? siRNA is what mechanism here. siRNA gene silencing. siRNA entered the cell via lipid-based transfecting agent. Okay, siRNA. Then siRNA binds and it make RISC, RNA induced silencing complex. So RNA induced silencing complex equal it will make. Then this siRNA stand for separate, activated, go inside the cells, and then what happen? After going there, they challenge to the mRNA, okay, target to the mRNA and cleavage the RNA and destroy. Like a building, if you destroy the building, okay, it's something will be there, the property will be there. No. So it's go here in different pH. I have told different pH range they will be crossing. Okay, they will be making RIS. Then RIS will challenge the siRNA, mRNA, and it will destroy. Okay, it will break like this. Okay, so we have shown these things. This figure is been taken somewhere, but we have done with this nanocapital. These things we have published in Nanomedicine. Okay, now how see? So whether it has gone or not, you can check. This is the cells. This is the cytoplasm. Okay, black plot. Okay, actually it is metal tagged. Okay, with metal tag, this nanoparticles going in the nucleus and the cytoplasm. It is means that they are going inside the cells. Okay, so normal cell size after challenging, I will see that how the morphology changes. It is means that they are clean. Okay, now you say that I will be going to these things where these core cell materials, porous materials is used. The thalidomide baby cases actually it has been tested with some things. The genetical disorder, how a drugs can create and how you can actually concept has been given how you can make it. So it's come from this our idea come from the Nobel laureates, these three Nobel laureates where they mention the asymmetric synthesis method or chiral drugs. What is that? Thalidomide actually they have a chiral center. Okay, thalidomide. Okay, this is the structure. They have a chiral center. They have two structure. Let's say what two structure will be there? There will be R and S. Okay, R and S. R thalidomide, sedative, anti-anesthesia agent, and thalidomide A structure is teratogenic. Okay, this thalidomide actually used for the morning sickness during the pregnancy. Okay, but whenever thalidomide R structure used, okay, they give the sedation and anti-anesthesia effect. CIPET. But whenever they are S type, they actually interact with the DNA or with the fetal, and they actually create this type of genetical disorder. Okay, this news has come, and this actually invented by this lady, okay, Kelsey, who received this award, actually prestigious award, and she has been allowed to do this job up to the 94 years. Okay, 90 years, retired at the age of 90 years. Okay, this on in her honor. Okay, so these things. So what we have done here, so chiral block copolymers like this had been synthesized. Okay, like this glutamic acid, gamma benzoyl ester. So polyethylene glycol attached with this, and finally this will be there, which is in property like a protein. Secondary configuration it can have. Okay, the chiral structure can be generated like this. You can do the chiral structure analysis by the CD. So if it is chiral, this will give the different spectrum. Fine. So what we have done actually, we have taken the silica, okay, and then we allowed the formation of the micelles of the chiral block copolymers, which deposited on the surface. Then allow the polymerization. Again, polymer they are deposited. So what is that? Nucleates or core, and then polymers form and it is deposited on the surface. Okay, polymer form and deposited on the surface. So that's the chiral. So hollow chiral. Fine. Then remove. Then pore is generated. Pore will be generated in the thin network structure actually, and which is actually the stereospecific in nature. So when stereospecific nature is there, when removed, this will be creating this type of porous structure. Pore, and these pore are actually has been tested solid network structure by solid state NMR. Okay, and what about the CD method? Or solid state CD method are there. Pore are chiral in nature. So that how it is, this is a porous structure. Inside this pore, all cases there will be core of the micelles. Okay, core of the micelles, and network of the polymers will be forming. If you taking silica or polymers, here it is showing polypyrrole. Okay, polypyrrole it is showing like this. So hollow core is in every cases you can take. This is the chamber. This is a chamber. Chamber have the network structure and hollow core. Okay, in the long sense, in nanoscale. Okay, this will be there. Then actually chiral drugs can be loaded inside this. Okay, chiral drugs can be loaded inside this, and it can absorb. It means that, okay, it means that a specific chiral, if it is L imprinted, L chirality imprinted, then it will be loaded with the L chiral molecules, and if it is D imprinted, then it can be loaded with the D only. Okay, so whatever the problem was there for the thalidomide cases, okay, thalidomide cases D and L. So if you need only the R, then you can imprint with the R, and D can be loaded, and S can be refused. So S will not be there. So specificity, whatever in the first slides I was showing, the memory effect. So this hollow core structure or core structure can be generated, okay, with the chirality, where the memory effect, it can memorize the particular structure, particular chirality, and this can be loaded. Okay, so I hope that you got this idea. Okay, what I wanted to tell, that these chiral molecules, specific chiral molecules can be loaded and it can be released. So like the thalidomide cases, you can remove these things. Let's say there is schizoplex, is another type of pore shell structure which is mesoporous in nature, where inside the porous we have taken the core photonic crystal zinc oxide. Okay, zinc oxide have defective network structure. Okay, it create the oxygen defect. And silica was the core. Silica is considered as the inorganic polymers. Okay, inorganic polymers inside the cores are there. What actually happened there? So we have, whenever we have taken the DNA, ETRB loaded DNA, it has found that inside these pores, DNA can be loaded if the core zinc are there. Okay, how it is happening is that, so there is a silica network structure, pores are there. Okay, pore which is not functionalized with the zinc oxide. Okay, then zinc DNA will be negatively charged and silica will be negatively charged. There will not be any binding. But if you take, let's say, pore and silica are there, which is surrounded, okay, which is surrounding surrounding the pores, then so a lot of negative charge, okay, and positively charged will be generated, and let's say DNA will be negative charge, DNA can be loaded. These cases, if you take this zinc oxide inside the pores or hollow structure, then lot of positively charged will be generated, DNA will be negative, and DNA will be can be loaded huge amount. So this is the scientific idea, actually basic idea. So if we understand the scientific background of making all this structure, then our things will be easy, which drug will be loaded and which cannot be loaded. Okay, so what actually happened is that we have loaded the DNA molecules and we challenge, okay, with the lymphocyte, okay, and K562 cells. Okay, so it can use for the leukemia. Okay, leukemia. And now you see, you can make hollow core, triangular core cell, okay, capsule type, rod core cell type, and let's say ganglion capsule I will be showing. Okay, now where is this state? Okay, so let's say polymers core like this, you can cell assemble, you can make this triangular core cell where the core is will be solid. Okay, these all are the structure. This is the core structure. Okay, let's say where the surface morphology is different. This is hollow, this is will be channel, and this is like a capsule which have a structure of like real capsule. So this is the core self-assembled inorganic. So zinc oxide, you can also tell this polymerized nanoparticles. Okay, how see, there will particle assemble, there will hollow. Okay, there will diffusion objective mass. So why it's created. Okay, go fast. Okay, so after 11. Okay, so you can make loaded the anti-cancer drugs. You can load the DNA molecules or fluorescence molecules, and actually you can study the release, different sizes of the particles, how that there will be zinc are there, negatively charged drug molecules will be attached on the surface, or it can go to the channel. Okay, so let's say doxorubicin has some structure, negatively charged, positively charged zinc oxide will be there. Okay, this can attract. So a lot of physical property, actually paramagnetic in nature. Okay, that property also we can. So these all are the characterization method. I'm not going through that, time is not there. Okay, now see polymer cargoes, let's say polymers. Okay, you can load, you see here whatever I have shown. Okay, solid core, porous. Okay, porous cells, you can see the white pores. Okay, you see again very clearly. Okay, loaded, you can challenges cancer cell, leukemia, okay, liver cancer cell, when it has been found that it has specifically attacked to the cancer cell. A lot of images are there, you can take it. So cell division you can actually restrict by this way. Okay, so cellular interaction whenever are there, okay, they kill this, they destroy the morphology. This is the, let's say, confocal microscopic images. Okay, you see that how they are actually killing. Now another one is that the virus made core cell polymer network structure. Okay, virus are there, it is the Quia mosaic virus infected. Almost size is the 32 nanometers. So cavity can be created equivalent size of the virus. Okay, so that is the network structure of the polymers, or whatever you will have, the inorganic materials are there. So that also we have. So you see that in high resolution images. So there will be cell, there will be core. We have used these things, so loading the various anti-cancer drugs, and you have used leukemia and some others. Okay, you have specifically one. So cancer therapy. Again, you can go, let's say, inorganic materials you can take. Okay, now another thing is that if it is this type of, let's say, so many hollow cores are there, not only one hollow, 3-4 hollow can you will make. Okay, or like a porous you can take here. Okay, immunologically drugs we have tested like OVA. Okay, and you have tested this can be used at the vaccine purposes. Now let's say another one I will be telling this type of things, polymers, N-phenylalanine methyl ester alkyl chloride. These polymers, it will be creating this type of hollow core. You see that. Okay, it has the specific property. Okay, if it has a specific property, we have, you can load. Not, I told that can be gas, can be there. Nitric oxide can be. Nitric oxide actually immunomodulatory drug. Whatever immunomodulatory drugs you can inject inside this body, and you can. So if you can load 5 to 18 and picomolar, okay, and you can deliver this, it has huge importance. Okay, remarkable discovery in modern medical science is NO, which long been regarded as a toxic element and pollutant, but it has play critical role in physiological processes. Okay, So mediator for immune system and inflammatory response you can control. Okay. Those who have the possibility of the heart attack, but blood coagulation properties, that cases also it can be injected. But you see that what major use, these are the structure actually. Time is not there, we can go fast. Okay, so we have tested various cases where this is going inside the cells. Okay, how increase the, so we have tested how it has released. I can go final slides. So where this can be used, hypertension treatment, vascular and obstructive diseases like a stroke, myocardial infections can be used, and inflammatory disease purposes also you can use these things. Okay, so you can, if you can process this properly, this core cell type of polymer material, where the polymer use some monomers, acrylic, cross-linking use, and this is like this. So it has function, pro-inflammatory function, immunostimulation, tumor suppression, and tissue injury. So a lot of uses will be there. Okay. Now, how you can functionalize? Okay. So you will have a core. There will be ligands. You have to attach the ligands, the core and ligand. Core acts as a molecule. So many cores you can take. Then cell you can make it different materials. Okay. And dopants you can use. Lot of dopants you can use, which actually increase or decrease the photonic properties. Okay, and let's say outside will be polymer. This can be concept. Okay, you see that inside the core that is these things, and outside these cells are there, polymer cell. Okay, you can make in any way. One example is giving the ligand. You can use the organic ligand exchange method will be there. Let's say ligand oxidation. Okay, whatever will be there, you can oxidize the ligand, and ligand attraction. So there is a core, ligand will be attracted on the surface, whatever I have told. Let's say polymer network are there which will be creating, or core will be there, on the surface polymerization will occur. Layer, when the surface polymerization will be occur, so on layer by layer deposition will be there. So this is inside the engineering, what is the science behind are there. So how you can make these things. Okay, now see, there is a core, there is a cell, there is a pores. You can use the anti-cancer drugs or whatever the photosensitive drug. Then you can radiate these things. Whenever it will be radiation will be giving, then it will produce the singlet oxygen. Okay, when it gives the singlet oxygen in different aspect, this singlet oxygen, when they are inside the cells, that singlet oxygen actually react with the DNA molecules and other organic components of the cell and destroy the cellular activity of the infected cells and kill the cells. So these are the concept. So what actually happened? So light will be there. Let's say photosensitive diode will be there, de-excitant diode will be there, and when in presence of oxygen, oxidizing radicals will be forming. Okay, oxidizing radicals actually can be non-critical oxidative damage, so there will not be any oxidative damage, and critical oxidative damage will be there. So if the intensity of this light is specific and it creates the radicals, okay, then oxidative damage will be there in cell death. Then what is happening? Schematic presentation. This is actually coming under the photodynamic therapy. So light plus drug, activated drugs, drug is activated, then oxygen, oxidizing radicals will be forming, the oxidative damage will be there in the cell. So, but these drugs you can take inside the cells, whatever I am telling. So here, inside the cells, okay, of the core cell structure. Now, so real cases, core cell, amine functionalized, you take lot of things, you attach the photosensitive molecules, okay, you can take photonic crystal, it can use for the therapy purpose, it can use for the imaging purposes, okay. These cases, the core are there actually upconverts and nanoparticles. I have not shown because to reduce the length of the stop, okay, almost we have finishing in 2 minutes. I will be finishing these things. Okay, then what is that? This has challenge against the quantum dot, how it's give, and this core cell materials how we give. Okay, it has quantum dot is toxic actually, but if you coat with the polymers, then what will happen? This type of toxicity can be reduced and you can use for the cell imaging purposes. So cell imaging and therapeutic all together called theranostic. So these core cell type of materials can be used for theranostic purposes. Okay. No conclusion, financial assistance, DST, SERB, UGC, lot of financial assistants are there. Okay, acknowledgement, many work are there from the PhD student, they have contributed. Okay, and yes, many of them are still working. These are the collaborators from this. And thank you very much for listening to me. If you have any question, I hope I have finished in time. If you have any question, okay, doubt,
Thank you very
you
much,
can
sir.
ask me now and you can mail me.
Thank you very
So,
much, sir. Thank you very much
okay.
for this extensive lectures on polymer, porcel, nanoparticles, and their applications in biomedical. There are a lot. I have seen in the chat box there are a lot more questions from the participants, and any participants, if want to interact with sir directly, please raise your hand. Mr. Z. Kiribakaran. Please unmute.
Sir, good morning, sir. Professor, good morning, sir.
Ah.
This is Krubakaran, sir.
Yes, good morning. Good morning.
Yes, sir. In the initial slides, you explained about the organic and inorganic materials, sir. Actually, I am doing research in polyamide, sir. I want to know how to categorize the polyamide. Is it organic or inorganic, sir?
Polymer is organic.
Polyamide, polyamide.
Polyamide is organic.
Yes, sir. Yes, sir. That's my doubt, sir. Thank you, sir.
How we define organic? Organic materials usually made up with carbon, hydrogen, oxygen. Okay, carbon, hydrogen, oxygen, and nitrogen. And inorganic, like silica, okay, zinc. Okay, these are the inorganic materials.
Sir, one more question. Polyamide is a degradation one.
Polyamide
Is a degradation one.
So if you make cross-linking one, that is rigid, and if you make straight chain, then some part can be degraded. Polyamide usually not degradable.
Yes, sir. Yes, sir.
Yes, degradable.
Thank you, sir.
I think there is a question from G. Kiru Pakaran in the
Can you please present the topic little slow? I'm just reading the question. Can you please
how we define organic or inorganic polymers? Because few polymers are organic.
Organic polymers are made up of carbon, hydrogen, oxygen, and nitrogen. Mostly. And inorganic polymers are, let's say, silica, Si, Zn. Okay. One example is silica, SiO2. But silica also tell organic sometime. In the middle of organic, inorganic.
Sir, there is other question. Is polyamide is degradation one?
Polyethylene glycol, let's say it can degrade. All protein, amino acid based, some polymers, they can degrade.
Sir, is there any other questions?
Thank you, sir. Kindly provide your email ID. On the first slides, my email ID is there. Okay, or you can search my name. You will get, with the name, you can search, you will get my email ID and phone number. Okay, kindly provide your email ID. Okay, fine. Can you please share? You just type my name, you will get lot of paper. I just copied and paste my papers. Okay, and many things is going to under research actually. So high at present I cannot share the slides. I'm very sorry. Thank you.
But in the link they can get
which
the
polymer will be the best for nasal drug delivery to the mucus? This is, I do not know. Acha, which polymers will be the best for nasal drug delivery to the mucus? But you have to take some materials which will not block the pleural or the, what about the oxygen passage is there? I just forgot the name. That should not blockage. Okay. So that if it's create the blockage, then what will happen? Oxygen will not pass. Okay. So this answer is not there. You have to think it. What about role of polymers in drug design and discovery? It's a huge. nano drug discovery, polymers have huge importance because polymer structure is similar to the human body. Human body are made up with polymers. Okay, made of protein, peptide, muscles are made up of polymers. So since it is a similar type, so it is useful. Polymers are used for nano drug delivery system. Okay, Radhesam Sharma has the question. Okay, any more question?
Sir, I just want to know, sir. In the drug deliveries applications, we use core cell polymer nanoparticles. Sir, can made mixing of suppose like normally what we do, we put the silica at the core and gold or silver at the cell. So what are the other alternative, those type of combination materials, sir?
So advantage of silica is that you can make various sizes of particles. Okay, bigger, smaller. In case of metal, I was telling in some slide actually, where polymer 1 will be depositing, can be deposited in polymer 2 or not. Just a minute, let's take these slides. I was telling in these slides, the polymer 1 can be deposited in polymer 2 or not. I was telling polymer 1 can be deposited on polymer 2 or not. So that is depend on the surface property. Okay, surface chemical properties. Now for the, let's say, silica and silver nanoparticles you can take inside the silica nanoparticle. There is no problem. Okay, but getting the silver nanoparticles in bigger size, let's say 100 nanometer, 200 nanometer bigger size. Okay, you can get. Okay, your question was there. You wanted to take in the core silver and silica on the cell. Okay, usually cell materials come with the smaller sizes and core materials come with the bigger sizes. So if you can reverse the size, then silver will be coming on the center and silica will be deposited along with the polymer network structure. Dr. Judika, are you correct? Are you clear?
Yes, sir. Yes, sir.
So usually core come the larger sizes of particle, okay, and cell along with the cell smaller sizes of particle. This can be happen for the polymers, where polymers network are there and two type of particles here is. But
yes,
if you take inorganic materials, then let's say silver and gold, both have the different in redox potential. Okay, usually what happens, which will have higher redox potential, that will be there in the center, and which will be their lower redox potential, that will be deposited on the surface. Okay, these cases, let whatever I was telling in the triangular core shell nanoparticles, I have not shown triangular core shell nanoparticles, where core is actually particular type of materials and cell is something different. These cases, I was taking the cell materials F7. You see that there is a inside some core are there and cell are there. These cases it is not possible because their redox potential are different. So balancing redox potential will be there. Okay, if redox potential is low, then it will be coming on the surface. If it is high, it will be there in the cell, in the core. Okay.
And sir,
now next question.
Last means I think. So what about the interfacial polarization effect of interfacial polarization? Just like core and cell is there, what effect it can play in the drug delivery system?
Effect can play the drug delivery, where I have told that surface functionalization. I have told somewhere surface functionalization. I was showing somewhere. Surface functionalization of these here. Let's see. Here I was telling the surface function. Here I giving the example for this silicon. Here you see that actually plus or minus it is means the polarization. Okay. Plus or minus means in electric field. What usually happen in polarization? If you have a materials, you apply the electric field. The separation of charge will be there. One side will be positive, then will be negative. So surface polarization will be there. So polarization. See, this is also one type of polarization. Okay, positive charge will be there, or negative positive charge due to the zinc will be there, and negatively charged will be attract the positively charged. So polarization will be having some role for loading the drugs. No doubt. Surface charge is very important.
Yes, sir.
With surface charge, it will be creating hydrophobic or hydrophilic, that thing. Any question? More question?
Sir,
I'll be
there
happy
is
with
one
this.
question in the chat box.
Uh-huh.
Will the polymer material can be used for biomedical imaging?
Yes, it can be used for biomedical imaging. It can be used for the biomedical imaging. Why not? The last slides were there. Imaging. Last slide. This is, I was telling. Okay, these cases where this you can use for imaging. And next slides, I was telling, you see that. Okay, it can be used for the imaging purpose. You can load the polymer particles with the fluorescence dye. Okay, which will carry to the surface, or photonic crystal. Okay, the carrier will be the polymers. So this can be used for photonic. Okay, recently probably we have communicated one paper where we mentioned the theranostic application, where the polymeric constituent is the carbon. We have the polymeric constituent is carbon, polymerization of carbon. Everything is carbon network structure for polymers. Next question.
Is there any
Good morning,
queries?
sir.
Good morning.
Sir, my question is, what is the role of ligand in polymer?
Ligand is the functional group.
It is as a functional group. Okay,
Ligand
sir.
is the functional group. You can functionalize these things. Whatever is
Okay,
slide is
okay, sir.
visible. Ligand and the functional group. Yeah, here.
Yes,
You see.
yes, yes.
Third example. Ligand is the functional group.
Ji. Thank you, sir. Okay, sir.
Okay. So I told that I will put the functional how you can functionalize in the last slides. Okay. The solid core network structure polymers. Then you can functionalize with whatever you want. Inorganic, organic. Okay.
Yes,
So by
yes.
attaching these things, you can change your property of.
Yes, yes, sir. Thank you, sir.
So okay.
Thank
Okay.
you very much, sir. Thank you
Thanks
very
a
much
lot.
for this illustrated and elaborative details of drug delivery mechanism through core cell nanoparticles using polymer materials, polymer composites. So it is a very advanced area of research, and we thank you very much. I, on behalf of CIPET CSTS-GUWAHATI, thank you very much for giving us your valuable time and sharing this information with us. We are looking forward, sir.
Thank you,
sir.
Juthika. Thank you, CPET team, for inviting me, and thank you all the audience to listening to me. I do not know how much I have able to.
Sir, 116.
116. Okay, all 116 participants. Okay. Dr. Anupama raised the hand. Dr. G. Krishna.
They have answered already. They have interacted.
Okay. Thank you all. Thank you all. At any time you can just communicate
Yes,
to me. Okay.
Thank you very much, sir. Thank you.
Thanks a lot. Thanks
Yes,
a lot.
Sir. Yes, sir.
Now I am.
You can leave the meeting.
Stop sharing and okay. Thanks. Thanks a lot. And leaving the meeting. Okay. Thanks. Thanks.
Yes, sir. If participants has any queries related to feedback, attendance, and test, they can message here or they can ask.
Excuse me, ma'am, there is any link for the feedback daily basis or at the end of the FDP?
Yes, ma'am. After end of the FDP, in the Author portal, the feedback link will be there. So you have to give the feedback there.
Okay, ma'am.
Yes.
Ma'am, one question is that regarding the test. So what is the timing for the test in the
So it
end
will
of the
be,
day?
yes, it will be on 11 February as per the schedule, and the test will be from 2:30.
2:30.
So
And what is the
I will
what is
inform
the time
you.
limit for the test?
I will inform you on Thursday morning whether it
Okay.
will be Google form or how many questions will be there, marks
Yes,
will
yes.
be there. And the PPTs, those speakers who have allowed us to share their PPTs or any other study materials, I will share at the Author portal by Wednesday.
Okay, ma'am. Thank you so much.
Yes, and attendance is compulsory, and since I think since all the speakers are working extensively on this topic, so I think you will definitely attend all the programs. So I am not speaking the attendance, but just speaking in that you just in on time in the morning session. Please try. Okay, so our next session will be from 11:45. And that will be delivered by Dr. Garima Agarwal from IIT Mandi, and she will talk on smart polymeric micro nanomaterial for controlled drug delivery. So now tea break is there, so you can be on the same line or you can leave and rejoin again at 11:40. Thank you so much.
Hello.
Good morning, madam.
Good morning, Dr. Jyotika. How are you?
Yes, fine. We will start after 5 minutes.
Sure, sure. No problem. I just wanted to check whether the screen is being shared
Yes.
properly. Yeah.
Yes.
Okay.
Yes, it is visible. Sharing.
Okay. But I was just wondering how I can stop sharing. Okay, here it is.
Stop
Fine.
sharing.
Another thing.
Yes.
Yeah, I could see it. Another thing is I see that you are recording the meeting, so I was just wondering, like, so you can record it for your own purpose, but please do not post it on the YouTube channel or something.
Okay. Actually,
Is the slide
as per the rule of AICT Auto Lab DP, we have to upload the session link. So by the software, we are recording so that we can submit. to
No
them.
problem. No problem. You can keep it for your own record.
Yes,
Sometimes people post it on YouTube or something, so that's the reason.
Yes. Okay.
Is the slide moving? Can you see the presentation moving?
Yes, it is moving. Now it is showing research profile. Yes.
It's
it
the
is
title
moving.
slide, and now it is the
Yes.
research group. Okay,
Yeah.
fine. So our audience today, it is, can you just tell me about the audience?
Yes, audience are all are faculties, scientists, researchers, means few researchers working in some research organization. Then most of them are faculties from different institutions of all over the India.
Okay.
Universities or different. And they are of different discipline. Some are from nano, some are from chemistry, some are from physics, then some are from biotechnology, some are from pharmaceuticals, like that. Some are from
Okay, okay.
mechanical, interdisciplinary type.
Okay, okay. So this FDP is for 2 days or it is only for 1 day?
No, it will be for 5 days.
Okay, can you share the program with me?
Yes. Program schedule.
Yes, I mean, if I'm interested, I can.
And yes, and that link is same for all the 5 days.
Okay, fine, that's great.
If at any time, if you got time,
Yeah,
can please join.
yeah, sure. So how many participants this time?
120.
That's very nice.
Yes, earlier one was elementary one, and
Yeah.
AICT had given us the advanced one of also that same topic.
Okay.
Okay, we are going to start. A very warm welcome to Dr. Garima Agarwal, Madam from IIT Mandi, for this expert session. Before starting the session, I want to introduce ma'am shortly, in short. So she is the assistant professor in School of Basic Sciences, IIT Mandi. Madam has research expertise in functional polymers and nanomaterials, synthesis of novel micro nanomaterials, design of interactive materials with active properties for targeted application, controlled release, plant care in agricultural applications, active packaging, functional coatings, biomaterial, drug delivery, and tissue engineering. So she is the recipient of different honors and awards. She is the recipient of prestigious DST Inspire Faculty Award by Department of Science and Technology, Government of India. Magna Cum Laude Award in PhD from the Faculty of Natural Sciences from RWTH Assam University, Germany. She is the recipient of DART Scholarship, Council of Scientific and Industrial Research Fellowship, and many other fellowships. She has a number of international publications, and where she has done extensively worked on these biomaterials for different medical applications. So we are very happy that Dr. Garima Agarwal ma'am had agreed to share this session with us. Ma'am.
Thank you very much, Dr. Jyotika, for the kind introduction. Let me share my screen. Please confirm once you see it.
Yes, it is seen.
Is it full screen?
Yes. It is.
Okay, thank you. A very warm good afternoon to everyone. And today I'm going to talk about, hello?
Yes, ma'am, audible.
Okay, yeah. So a very warm good morning to all of you. And today I will discuss about smart polymeric nano micromaterials for controlled drug delivery. Before I discuss this scientific part of this talk, let me first tell you where I'm coming from. I am working at IIT Mandi, and this is our South campus at IIT Mandi. So we do have two campuses. One is South campus, one is North campus. This is the view of the South campus. And if you move around this mountain, around 10 minutes by bus, you will have the North campus. So here is the sports ground. I'm currently sitting here giving this presentation. Here are the faculty offices and the administrative building here. The round buildings that you see, we have the signature mess over here. So whether you go to the North campus or the South campus, if you see a round building like this, you know that it is a mess dining facility. We do have library here, also one library in the North campus. And here are some of the staff accommodation. Some of the faculty accommodations are still under construction. These are also the faculty accommodation. And if you go further here, you have more faculty accommodations here. And this side, here at the bottom, you have Pool River flowing here. So basically the campus is all surrounded by mountains. So if you ever get a chance, please do visit us and I'll be happy to host you here. So I'm currently working at IIT Mandi. Before that, I was working as a DST Inspire faculty in the Department of Polymer and Process Engineering. I did my postdoctoral work at Ghent University, Belgium, and pursued my PhD in RWTH Aachen University, Germany, in the area of polymer chemistry. Before that, I did my MTech in Polymer Science and Technology from Indian Institute of Technology. So with this short background in the area of polymers, let me go ahead with the presentation today. So this presentation is divided into 4 components, major 4 components. First component is the introduction, where I will tell you about the microgels, which is the first component which I have used to design different systems that we will be discussing today in this presentation. Second component, can you please mute yourself? And the second component is polyalkoxysiloxane, which I have used in combination with microgels to design different systems that we will be discussing. The second component of the presentation is functional microgel. So basically, how we can use or how we can design microgels with different architecture, different chemical functionalities, and how we can use them for release of cancer drugs or release of different other drugs like aspirin. The third component will be the synthesis of capsules by using both microgels and polyalkoxysiloxanes, and how we can design these capsules by using these 2 components with the help of interfacial hydrolysis and condensation method. The last example that I will be discussing today will be the synthesis of natural polymer-based nanocarriers for drug delivery. So basically, I'm going to give you the examples from both synthetic polymers and natural polymers, how we can design different systems using these polymers. So first of all, why the functional polymers and nanomaterials are so important? So we know polymers have various attractive properties. We see polymers every day in every daily activities, the chair we are sitting on, the laptop we are using, the computer, desktop, many parts, the water bottle, even if you are drinking water right now, or anything you can imagine, we in some way or the other, we are using polymers. Whether you go to the market to buy something, then also you are using the polyethylene bag, which is also polymer. So polymers can be of different varieties and they can have different properties. May I request you to please mute yourself?
Yes, ma'am. We are checking who is.
Thank you. So polymers can be of different properties depending on how you synthesize them, what synthetic strategy you are using, what monomer you are using, what co-monomer you are using, what reaction conditions you are using. So depending on that, the polymers can have various properties. For example, they can be soft colloids, they can be responsive. What I mean by responsive is that they can respond to different stimuli. They can be biocompatible depending on what type of monomer you are using. They can be porous. They can have different chemical functionalities. So whether it can be amine functional groups or carboxylic groups or different functional groups, even at the advanced level. And they can